to welcome everybody to the October Museum Talks, Snapshots of Biodiversity, stories from a 10-year Bioblitz survey effort on Vashon. I'm Elsa Kronquist, the Executive Director of the Heritage Museum. And this is a reminder that your microphone is muted for this talk. And if you have any questions, please type them into the chat function and we'll answer questions at the end of the presentation. I'd like to take a moment and say a very big thank you to our sponsor, For Culture, for sponsoring our Museum Talk series. The next slide, please, Chris. At this time, I will introduce Chris Austin, who is a Heritage Museum board member, co-host for this museum talk, and host of our radio show, History Worth Hearing. Chris will acknowledge the first Vashon Native people through reading our land acknowledgement statement. Chris?
Thank you, Elsa. Before I do the land acknowledgement, I'd like to talk a bit about the person on the screen. Her name is Lucy Duran, and she's a very important figure in the history of our islands. She was born on Vashon in 1843, and her testimony in the 1927 Land Claims Tribunal provides the most complete information that we have about the life of the Shobop's Native people on Vashon. She died here in 1929, and she was buried in the Vashon Cemetery, but her grave was unmarked until 2008. That's when a headstone was placed on her grave in a ceremony that included the Puyallup tribe as well as islanders. So let me get to the the land acknowledgement. The Vashon Heritage Museum stands on the ancestral lands of the Shwabop's or Swiftwater people who inhabited Vashon Maury Island for millennia. The Donation Land Claim Act encouraged large numbers of Euro-American settlers to claim this land, even before the Medicine Creek Treaty of 1854 extinguished Native claims to it. The Shwabop's were forcibly removed and relocated onto nearby reservations, including Puyallup, Nisqually, Squaxin Island, and Muckleshoot. We acknowledge the people of the Puyallup tribe whose stewardship of and relationship with Vashon Maury Island continues today. We commit to amplify the voices of Indigenous people, making space for them to tell their own story, both past and present, in our exhibits and programs. Back to you, Elsa.
Thank you. And next slide. So now the Vashon Heritage Museum Inclusion Pledge. Next slide, please. The Vashon Maury Island Heritage Museum rejects racism, discrimination, and hate. Our purpose is to honor stories of love, oppression, action, and resilience. Stories that give voice to all. Examine hard questions about social, economic, and environmental justice, and reveal racism and oppression within our heritage. We pledge that everything we do will honor the island's diversity of cultures and histories. Next slide, please. So the museum, this will be the last talk for 2022. But that doesn't mean the museum will not be busy, because we will. So we will invite you to, the museum will be open, and we invite you to visit Natural Wonder and our new Audubon mini exhibit that will complement Natural Wonder, which features birds and songs of Vashon, that will be open in in time for the first Friday in December. And then we'll kick off the new year in January 2023 with a new series of 6 Vashon 101 talks. We're bringing back the faculty from the Vashon 101 class as a museum talk series. So watch for more information in our newsletters and sign up for the talks in December. And Chris, would you like to introduce this evening's speakers?
Yes, thank you. We're very fortunate tonight. We have 2 great speakers. Our first one speaking will be Bianca Perla. Dr. Perla is the director of the Vashon Nature Center, and she will be followed by the doctoral student, Andrew Simon, who is also the curator of the Biodiversity Galliano project. So Bianca, I'm gonna let you take it away. I will stop sharing and you can begin your lecture.
Okay, thanks, Chris. And thanks everybody for coming. I'm going to pull up my slideshow here. There we go. And thanks, Andrew. Also for doing this talk with me. Andrew's going to patch in at the end and take it far beyond the local scope that I'm going to be talking about in the beginning. So what I'll be talking about is the 10 years of exploring island biodiversity through Bioblitz events. And I'll be highlighting stories from this effort, and also talking a little bit about the human community that's been growing around these events and spinning off from them. A lot of the information has ended up in the Natural Wonder exhibit, so I encourage everybody to also visit Natural Wonder at the Heritage Museum if you haven't already. It will run through June, and we've also through these events made connections not only with each other and our community on the island, but also with a wider network of people and different communities who are doing these same types of events all over the Salish Sea and beyond. So we'll hear a little bit about the wider scale implications of this type of citizen science research from Andrew Simon at the end of this talk. So first, what is a Bioblitz? A Bioblitz was started way back in 1996, and it was started in Washington, D.C. through the National Park Service as a way to try to give people an idea of what biodiversity meant. And so Bioblitzes are 24-hour species marathons where scientists, naturalists, and members of the public get together and count as many species as they can in a 24-hour period in a certain particular location. And so since 1996, these events have spread all over the world. And our first one was in 2012 at Neil Point. One of the reasons that the Bioblitz events was created was that the term biodiversity and the whole concept of biodiversity is a super important concept to the functioning of this planet and our life support systems as humans. Yet it's highly threatened today. Biodiversity is declining on a very rapid scale. Yet it's a really hard term to internalize emotionally and intellectually for people. So it's a hard concept to sort of rally around. And so Bioblitzes were created to give people a real firsthand understanding and experience of the biodiversity that exists right in their homelands, right in front of their faces every day. And so biodiversity really comes in many different scales, from genes, it's just the variation in life from genes to species. And Bioblitzes tend to focus on the species level of biodiversity to ecosystems, and doing multiple bioblitzes like we have on the island gives us an idea of that variation in ecosystems, which you'll see throughout this talk as well. So this biodiversity, this variation in life is really the crucible for life, and it's really the creative force that keeps life going and changing and adapting through time. And we depend on that for lots of different services, from decomposition, little bugs that are decomposing our garbage, and building our soils, to pollinators that pollinate our food. That diversity is supporting us all the time, and it's a very important part of our connection to life. In a Bioblitz, the main question is, how many species can we find in 24 hours? And so it's really just a snapshot of an area. And it's a high level snapshot of what's there. And so some things you might do during a Bioblitz are do a beach survey and turn over every rock you can find and put them back nicely, and find whatever you can. And then we also use traps. We use live traps. We don't kill anything in our Bioblitzes. We don't take any specimens, because we're also using this as a celebration of life. So we're honoring and respecting the life as we go about our surveys. And so we use live traps and we let things go. That's Gary Shugart in the middle of the pond, trapping amphibians on the upper right there. In the nighttime, we use night lights, black light systems that draw in bugs to a sheet, and we find out how many things are living around in the forests and the fields. And then we get up super early and we check our little live traps for mammals, small mammals that may have gone in them at night, and we get a survey of those mammals. So those are some of the things that we do in a BioBlitz to survey a certain area. And why are we crazy enough to do this? Staying up all night, counting creatures together? Mostly because it's fun, but also because we are in the midst of an extinction period that could be as big as the last 5 major extinctions in Earth's history. And that's, we want to bring awareness to that, and also kind of think about what that means, when we're looking out there at all this biodiversity. Where is it headed? 50% of the species on Earth is predicted from their population trends to become extinct in the next 40 years. So that's a huge amount. You can see here, these are the different amounts of species that have gone extinct, percent extinction of species in the last 5 major extinction events on Earth. And this is us right now and what's predicted. So part of this is bringing awareness to this major trend that's kind of invisible in day-to-day life. Another reason that we like to do BioBlitzes is because it's fun to really look at your home in detail, and to see the richness of life and appreciate the richness of life, and also come together with people who also appreciate that. There's something very heartening that balances that declining trend we were talking about in the last slide, that brings us together around our shared love for nature and allows us to witness the beauty and the richness of life around us. And that's quite empowering. And I think that's one of the most powerful aspects of a BioBlitz, actually, is that human community and just that respect and awareness of life that you get when you're doing these events. So one thing that we are doing when we're out there is we're just witnessing, we're just seeing these different animals and plants and the variety, and just learning some of their stories from the experts that are out there that know them and have dedicated their lives to really understanding them. So that's an incredible gift that we have given from our leaders in these BioBlitzes. So in the Vashon BioBlitz events, we've done 7 in the last 10 years, and you can see them here outlined on this map. And they have covered about 7 square miles total of the island. Our island is 37 square miles, and so that's almost 20% of the island. And you can see also that we picked places that were in different spots on the island, from the north to the south, east to west, to try to capture different environments that we knew of, the little microhabitats on the island. So we'll go through all of those. And you can see those differences a little more up close. This is a summary graph of all of the species that we have recorded so far, the unique taxa. So that's 1,715 species. We've had over 900 participants in these BioBlitz events over the years. And these, you can see that the different BioBlitzes are here on the x-axis going through time. And then you have bars going up, that the green part of the bar is the number of new species that we find each BioBlitz, and the orange or kind of tan part of the bar is repeat species. So you can see at Neil Point, green, it's all green because we hadn't done one before, so everything was new. In Shingle Mill, which was our next one, our Fern Cove BioBlitz, we recorded 129 species that we had also recorded at Neil Point, but then we recorded another 234 new ones. And so this is kind of interesting, because you would expect as we go on, we would be finding more and more species, and the repeats would get higher, and the new species bars would get lower. And that was happening for a little while, but now we're increasing again. So what this tells me is we're really not even close to categorizing all the biodiversity that lives around us, and we're almost at 2,000 species here. So that's kind of I don't know if you can imagine what that is, but that's a lot of life that shares our island with us. When you're going about your daily life, you probably see this is a breakdown of how our species fit in, proportion, all 1750 or 1,715 of them. So you can see a lot of them are invertebrates and plants. Then we have the mosses, fungi, lichens, algae, and the vertebrates. And so when you're thinking about your daily existence, these are you probably see mostly vertebrates, right? You're aware of the birds around you once in a while, maybe some of the plants as you're walking to your car or back and forth to work or school. But there's all this unseen diversity around us that's there every day. And it just takes either those experts that dedicate their lives to finding these things or an event like this to uncover them. So that's one of the beauties of the Bioblitz. I'm going to show you now where a lot of our data is stored. It's a program called iNaturalist, and we're creating a database also at an internal one for the Nature Center to be able to house all of these records too. But right now we have this project on iNaturalist, and iNaturalist is a really great platform that anybody can use, and it's a citizen science platform. And you can get an account, and anybody can upload an observation. And so we've created this Vashon Biodiversity project, and we've uploaded all of our data to it. And from all of our different BioBlitzes, you can see there are more species here than we have recorded in our BioBlitz, and that's because anybody who falls within the boundaries that we set around Vashon here, there you can see we have a project that collects everything in a kind of a triangle around Vashon. And so here are all the records for Vashon Island, including our BioBlitz records. And what's really neat about this is you can actually drill down, and anybody can do this, which is fun, and say, maybe I just want to see what reptiles are here. And so you go, and there's 207 observations. These are reptiles. You can see these follow a road. So these are all roadkill. That's from Gary Shugart that's done that. You can say, oh, we have 7 species. What are they? And then you can see what pops up. And then maybe you're interested, wow, we have rubber boas here. You can go in, say, let's see where those are. And it plots it on the map where people have seen rubber boas. And you can see right away, wow, they're all on Maury Island. And then you can drill down even farther and say, well, what's this observation like? And you can notice it's tagged as research grade. And that's the really cool thing.
Bianca, can I interrupt? I'm sorry. I don't think anyone can see what you're showing right now, probably because you're just sharing one window.
Oh, no.
So
thank you for letting us know, or letting me know. Here, let me see. Okay, let me stop sharing, and I'll share my entire I'll share this, and then you'll see everything. Now, can you see?
Yes, we can.
Okay. Is everybody seeing that? I hope so.
Yes, we can see it.
Okay, good. All right, so I'm sorry. Let's go back a little bit here. All right, so I wanted to show you guys the Vashon Biodiversity Project, and you can drill down into filters, which I just went to, and I said, I want to see just reptiles, but let me take that off. And then you can see all of the different observations, and those include our Bioblitz data. So anybody can come in here and and explore and see what we've got and see what other people have contributed as well. All right, now I'm going to go back to my PowerPoint slideshow. Can everybody see that?
Yes, we can.
Okay, great. So now we'll go on from there. Okay, so Vashon has a huge variety of microclimates and habitats within our 37 square miles. And you can learn a little bit more about this in Natural Wonder, but we have a rain gradient of 49 inches over here where I am in Reddings Beach area, all the way to Point Robinson, which is only 38 inches. So 11 inches difference when you go across the island. We also have a gradient of soils, more sandy on the south and more impervious as you go up north. And that's an artifact of the different glaciations that have come across our island over time. And so keep that in mind as we go, because you'll see some differences in the species that we get in different areas. And it's partly driven by this and also the topography of the island. So let's go on a tour. The first thing we'll do is go to Neil Point, which is in the south part of the island there. And Neil Point has some interesting topography. If you look close, you can see there's sort of a staircase type thing happening here, where you have flat, and then you have a steep cliff, and then another flat area, and then a steep cliff, and it steps down towards the water that way. So these cliffs are eroding over time, and they're eroding in this kind of step pattern. There's a rotating fault block here that's doing this. And what's really interesting is that on these little flat areas, we found some ponds at the Neil Point Preserve, and they were in 2012 they had no bullfrogs, so they were all native amphibians and aquatic invertebrates in these ponds, which was quite unheard of, really, for the island. And they're also some of the only ponds that are natural. Most ponds on the island are man-made or man-encouraged with berms and other things. So really cool little pond habitats there. The forests were not very diverse, especially in terms of lichens. We noticed as we went to other places that in comparison, these forests in Neil Point had really no sensitive lichens left on them and in them, and we're wondering if it has anything to do with it being right across from Commencement Bay, and possibly either being in a sarcoplume, or having also paper and pulp mill plumes coming towards it over time. The eroding cliffs interact with the prevailing currents to move sand northwards all the way to Jensen Point. And the eroding sand of this point also goes westwards to Tahlequah Creek, feeding the beaches all along the way. And so really, when you get down there to Neil Point, you have these beautiful sandy, flat beaches that are incredible habitat for beach life. And that's one thing that stood out in Neil Point. And we also happened to survey this before the sea star wasting disease epidemic hit. So we have the highest diversity of sea stars that we found in any Bioblitz here at Neil Point. And here are some of the photos from that survey that shows the diversity here that you don't see as much anymore. So Fern Cove was our next Bioblitz, and that's up in the northwest part of the island. Anybody who's been there knows it's pretty wet, and lots of ferns and maples and cedars, and also really amazing eelgrass flats in Fern Cove. And so that really stood out for this Bioblitz. This is a little brooding anemone that lives right on the eelgrass blades. So this is a blade of eelgrass about 1/4 of an inch wide, and these brooding anemones live on it. And here are the little anemones that they're brooding. These brooding anemones are either all female or hermaphrodite. There are no true males in brooding anemones, and then they pop out these little babies here. And these guys eat crustaceans in the eelgrass beds, and they're also eaten by by some nudibranchs, shaggy mouse nudibranch, and some fish and other things. So they're important parts of the food chain here in the eelgrass beds. And eelgrass beds act as nurseries for a lot of different things. Here's some crabs and fish eggs. They provide a substrate for fish to be able to lay their eggs on, and there are a lot of young fish that swim around and young animals that swim around in these eelgrass beds. And then we have predators too, like the sunflower sea star. This is one of the last years we recorded it. The populations have really decreased because of sea star wasting disease. But hopefully sea stars are recovering slowly from that now. The location of Fern Cove also acts as a migratory bird trap sometimes. And so we got lucky on this Bioblitz that we got a spotted sandpiper, which was the first record for Vashon Island. And he happened to come during our 24-hour survey, which was pretty cool. The uplands of Fern Cove, very wet, especially since it rained during our Bioblitz, but it's usually wetter here than other places. And so this is Maria Mettler from the Nature Center, and also Raina Holtz, who was a very important leader in a lot of these Bioblitzes, looking at a lot of different things. They're looking at the snail right there, I think, which is a carnivorous snail that eats slugs. So gardeners might want to have that one there. We also found a lot of different amphibians. That's a western redback salamander and an ensatina. And these are things that people don't, they're here, they're on the island, but you don't see them very often. But there were a lot of different amphibians in this Bioblitz that because of this wet environment. Now we'll go to 2014, Christensen Creek on the west part of the island. It's kind of, that's my watershed. Christensen Creek is mostly privately owned. And so we had generous landowners that let us do some surveys down there. And there's a big remnant old growth forest and some remnant old growth trees there. So we picked up some amazing things like Devil's Club, Indian pipe that exist in a few other places on the island, but here they're kind of more part of the understory than they are other places, and likely because of that old growth characteristics. One thing here we've got is a little tiny mystery that was found on a tiny twig by our lichen expert Phoebe Goit. And she picked it up because she didn't know what it was at first. She went home, looked under her microscope, and looked at all her lichen guidebooks, and still couldn't figure out what it was. There was nothing that looked like it. And so she sent it to us. We thought for a while, maybe it's insect eggs, maybe it's kind of fungi. We started fanning it out to experts all over the place. And finally, the answer came back from Cornell University, a mycologist that did recognize this as a fungi. It's a little tiny bird's egg fungi, and it's called Corticium mensae. And you can see this little cup here, and then there's like a little egg in the middle of it, and that's the sporing body. And so little tiny rain droplets fall in there, and the spores spread all over the place. So we looked up this plant and realized that there were no records for it in Washington State. So this is now in the herbarium at the UW as the only record of this in Washington State so far. So our little twig is famous. Another thing we found in Christensen Creek, there were a lot of springs, and in those freshwater springs, there were little spring snails. The three on the left in particular are the ones that we were interested in. These are Pristanicola spring snails. and asking, and you can learn more about these in the Natural Wonder exhibit as well, but asking the snail expert how they got here. Some snails hitchhike over on birds, and I said, could that have happened with these? He said, no, they would dry out and they can't desiccate and still survive. And I said, well, maybe they rafted over on saltwater, rafts of debris. And he said, no, they wouldn't be able to take the salt. They're very sensitive species. And I said, well, then how do they get over here? And he said, well, they probably came over when Vashon was surrounded by fresh water. And so that was, the last glaciation when we were surrounded by Lake Russell, which was a freshwater lake, is probably when these guys migrated to our shores. So, over 10,000 years ago, maybe some of the earliest island residents. So that was pretty cool. And now we're going to go to Maury Island, going from wet side to dry side. We tried to survey all of the preserves, the whole preserve network over on Maury. And as we saw in that rain gradient, we've got a lot of dry forest areas. We have sandy soils, south-facing slopes, and probably a history of fire that has created these madrone forests. And they're an interesting little microhabitat we have on Vashon that supports a lot of different things. And looking in here, this is Michael Laurie, who helped on the plant team and in the madrone forest. And then we had Harsey Parker, who is a really valuable part of not only our iNaturalist presence, but also all of our insects here in the Bioblitzes. She found and photographed, this is her photograph, native katydid, when we didn't even know we had katydids on the island. So that was a pretty interesting find. Another species that is only occurs over on Maury is the western fence lizard, and we were able to get him them recorded on the beaches and going all along the driftwood in this Bioblitz. Maury Island Aquatic Reserve also wraps around almost the entire Maury Island, and it's one of 8 state aquatic reserves. And the birding team on this Bioblitz found this marbled murrelet pair that was foraging off the shores in the middle of summer. And that was a really interesting observation, because that's during the breeding and nesting season of these birds. And these birds nest only in old growth forests. And so they were nesting, and they can nest up to 50 miles away from their foraging grounds. So possibly this little bird was nesting in Olympic National Park or National Forest, or way down in Mount Rainier old growth forests. They need large old growth patches for their nests so that they don't get predated on by the crows and ravens. And but they need seafood. And so this just drove home the point that the island is really connected to a lot of bigger ecosystems and an important resource for these tiny endangered birds. Judd Creek was another 2016 Bioblitz, and what really popped out there is that there are a lot of wetlands and meadows in the Judd Creek watershed. And so we picked up some new species. This is one that Jim Evans, our plant team lead, found, skullcap speedwell. It's an obligate wetland species, and we didn't have any records of it being on the island before this event. And interestingly enough, it was found in a small private wetland. So it was not in any of the land trust preserves or the King County protected areas. And so that kind of, drives home the point that all different lands are important, and we never know what we're going to find in little pockets of private land too. So that was pretty interesting. And then we also had we did a night insect survey at the Matsuda, what's now Matsuda Farm. It was still meadow at that time, and found a lot of different night flying insects, one of which, and these teenagers spent a long time photographing, and falling in love with this creature, which is a very common, it's a 10-line June bug. But I love this because it engaged these guys for about an hour and they just fell in love with a 10-line June bug, which is pretty common. But it's when you start looking at it up close, grabbing onto its little piece of grass and staring back at you, and when you notice those big, huge antennae that this is a male that he's using these to sense the pheromones of the female around. And these are agricultural pests. They eat the roots of plants and trees, and yet you fall in love with them anyway when you spend an hour with them photographing them. So that was a fun little experience for those guys. Our Tramp Harbor Bioblitz in 2018 was quite notable for a lot of new species. And I think one reason is we had all of Chautauqua students helping us because it included Chautauqua Forest. And then we also had just such a variety of habitats. We were covering both Ellis Creek and Ellisport Creek that run down to Tramp Harbor. And we sent a small mobile team of crack naturalists down all the way down Ellisport Creek to the mouth, and that was quite a bushwhack in some places, but we had people looking for insects and plants and mammals and birds, and they made an incredible team and got a lot of new observations there. And we also had a lot of really cool things that the kids found when you just have that many eyes. And that's the beauty of a BioBlitz, is that, you know, it actually sometimes people who aren't the experts are the ones that are finding things because they don't have a typical search pattern that they're looking for. And so one of the things that kids found in my group up in the forest was a coral root, for example, and that was right in the Chautauqua Forest. And these are plants that don't make their own chlorophyll. This is a little orchid, and it taps into the fungal network and squeezes its nutrients from that fungal network that connects the different trees and shrubs under the ground. And it's a beautiful, beautiful little flower up close. We also had a great variety of beaches, rocky, sandy, and the pier at Tramp Harbor, which gave us a lot of diversity along the beach, and the salt marsh, which we also found right up from the salt marsh in Ellis Creek. We were able to document a beaver who I think still lives there, and you can still see the signs. So it was a really exciting BioBlitz for a lot of reasons. This was also the first BioBlitz we used iNaturalist. So that was quite a grand experiment as well in many ways. So then we took a break for a few years, and we ended up doing Heart of the Island BioBlitz this year. And that is right smack in downtown. We decided that after surveying all of these different natural areas, we had the Natural Wonder exhibit going on. We just wanted to see, what is it like right in the middle of our busiest human occupied spot on the island? And it was pretty amazing, this BioBlitz. We had Hans Youngman come out, and he did an incredible job of documenting a lot of the planted trees. This is a beautiful birch right next to the Fair Isle Animal Clinic. And it's really neat to go through iNaturalist now and zone in on these trees. When I was doing that, I realized, gosh, there were so many of them that I've gone by so many times and not even noticed. So it's a really a neat cultural story of how we as humans like to keep these trees close to us. And you know, they might not be the native trees, but they're in such numbers that they're providing some interesting and unique habitat for terrestrial insects and birds, especially too. We also had a lot of native plants that were planted by humans. So this is a Pacific rhododendron in Fisher Pond area that was planted. We had the nice native plant garden that the Garden Green and also the Audubon both planted a ton of plants in the Heritage Museum backyard. And then there were also stormwater ponds in Roseballin that had some native plantings that increased our species list. And Alan Warnicki, seen here on the right, has done our dragonfly surveys and was pretty impressed with the diversity of dragonflies that we saw at Rose Ball, and we saw 11 different species, and then he counted a 12th at Fisher Pond, and together he said, 11 species is quite a lot for this small stormwater pond. He says he sees about 11 or 12 species at Mukai Pond in a day. So this is providing habitat, and you can kind of think it's just a stormwater pond, but the way it has been planted has provided habitat. This is a little very young damselfly here right in front of the storm drain. These pictures are taken by Catherine True. And then there just another beautiful picture of dragonflies and the diversity that they can hold. Then we moved to Fisher Pond, which is also, that was also in our heart of the island Bioblitz. And it is a pond that is man-made with a berm in the, it was a more of a bog, and now it's become a pond. And it also, we put a black light up there, and we got a lot of different night flying insects that came. This is just a snapshot of some of our Lepidoptera that we got during the Bioblitz, and many of these came to the many of the night flyers came to the tent, the sheet that we had up at Fisher Pond. And we also, through connections with Bruce Hallman, have gotten connected with a bat biologist in Australia who helped us track down some bat sonar detectors. And so those were fun to use at Fisher Pond. And this is the bat group waiting for the sun to go down. These are the pictures of the little devices that can then record the bat sound, and then that bat sound can be identified to species. And so that was quite interesting, because we found these recorders recorded 9 different species of bats at Fisher Pond, and that's incredible, because we didn't know that there were 9 species of bats using the island. And we knew of some. We knew from previous studies, the silver-haired bat, the big brown bat, the little brown bat, and the California myotis. But all of these other ones we had not detected on the island. Gary Shugart has a record of a Miller's myotis as well, which would bring it to 10 bats. There are 15 species in the entire state. So this is pretty amazing, the diversity of bats probably coming in for all of those flying insects. The 2 that we're a little shaky on, but we got songs from, but that we're surprised about, are these 2, the canyon bat and the western small-footed myotis. And those 2 do, all of the other bats are known in our Western Washington area, but these aren't. And so some possibilities are that the sonar isn't quite correct, or that these guys were blown in by storms, or that there are some migrations happening and animal movements due to climate change that we're seeing. And these are, we're starting to pick up these things in a Bioblitz. So there's a lot of questions that come up in a Bioblitz that we can't answer, but that also generate further research. Looking around, for example, at the iNaturalist records, the closest western small-footed observation is on the east side of Vancouver Island. So that makes you wonder, are they using the Salish Sea, that inland sea, as a migratory route? And that would be an interesting thing to look into further. So in wrapping this up and getting on to Andrew here, one of the really neat things about a Bioblitz is that even though you're looking locally, your community starts to expand, not only in terms of the people that you meet at these Bioblitzes across, many different ages, because everybody can do this. These are our fungi team leads for the heart of the Island Bioblitz, Ren Hudgens and Hannah Morisoff. And we also had a visitor that just popped by, Kurt Miller, who was a mycologist from Puerto Rico. So he joined this group. So you get these really interesting connections that go much farther than the island. We have connections through time, like Yvonne Cooperberg, who has held down our base camp, the first Bioblitz, single-handedly at Neil Point for the entire time. And then here she is again in 2022 with her bigger support crew and a nicer sign than a cardboard box. And then we have the team leaders that are acting as mentors for these younger people who are interested in nature, and also just as really great storytellers that are telling the stories of these species. And we have the kids that get a chance to grow up, you know, sleeping the night away and on these preserves and seeing, you know, getting up early and seeing the sunrise and seeing all the animals out at night at the black lights. And it's an interesting and beautiful experience. Quinn, the redheaded boy here, is now on his way to college this year, and he's going into environmental science. Alex, who went to our first Bioblitz, is just graduated from her grad school in microbiology, and she's doing DNA work in Seattle on plants. We just had a whole bunch of interns join us this summer, and they were able to learn from these team leads and help us set traps and do all of the hard work that comes with the Bioblitz. And so really a community is being made around these things, both within and without. And so there's a lot of things that we can think about doing next. Next steps, like a biodiversity atlas of the Salish Sea, and that Andrew is going to talk about a little bit, and just creative outreach about biodiversity now that we have all of this information, and then possibly more Bioblitz events. So now I'm going to turn it over to Andrew. Here's us here on Vashon Island. Andrew is patching in tonight from Galiano Island, way up here. And did I get that right? Hopefully I did.
Yeah, you did.
Okay. And so welcome, Andrew. He's got a few words to say that just take this from that real local level outward. So take it away, Andrew.
Thanks. I'm not sure if my video is going to pop up here for everyone to see, or if it's just my voice. Can everyone hear me?
I can see you and hear you.
Great. Thanks so much, Bianca. I really appreciate the invitation to be here this evening. It was a really inspiring talk. I think it's interesting, you know, when you live on a little island and you're totally engrossed in the biodiversity, it's easy to kind of get lost in it. Every once in a while, it's nice to get a reminder from someone who shares your passion, that you're connected with communities elsewhere in the Salish Sea who are doing all this great work. And I'll speak a little bit about it shortly, that there's, you know, a network of these different projects within the bioregion doing this kind of work, but it seems like every community takes a different approach. And yeah, I really just loved a lot what you shared, Bianca, about connection between what we know about the natural world and the living experience of the people who live in places. Yeah, I could go on for a bit, but
thanks
for inviting
Andrew.
me. I am Andrew. I was introduced, I think, earlier as a curator of a biodiversity project here on Galiano Island. That's how I got to know Bianca. It was some years ago I reached out, having founded this organization called Immerse, the Institute for Multidisciplinary Ecological Research in the Salish Sea. And so, yeah, it says a lot about Bianca as an individual who cares as much as she does about biodiversity in the region. She traveled all the way up here to participate in this event, which is a founding event, where we envision what this organization might look like. It looks like these slides are proceeding automatically. Yeah, I'll just speak briefly. Apart from curating a local biodiversity project, I also recently completed my master's, and I've been working on publishing my work. So there's some examples here of my research, and each one actually folds into it some element or aspect of community science. And so there's things that really reflect what Bianca shared earlier, especially that idea that, you know, we easily get the impression that it's the scientists out there doing the work, documenting biodiversity, but in fact, you'll find the same case time and time again. Really, when you look into it, we know a lot about the natural world because of the work of motivated naturalists, so-called amateur naturalists who live in places and dedicate their lives to it. So one of my papers here, again, it's gone back, toward an atlas of Salish Sea biodiversity. It's a recent one. We showed that about 60% of what we know about the local marine biodiversity is testament to the work of local community scientists who've been active in the region. That's the Pacific Marine Life Surveys. And I just gave a talk today about bumblebee species extirpation and colonization, and that's research that I was only able to do because I'd done this work of consolidating a historical baseline record of the historical bumblebee community. And it's sad, it's kind of a sad story, but we were able to detect that since the 1990s, we've lost half of the species diversity that was once here. So we went from 10 species to 5, and actually we have 6 now because of a recent arrival. But yeah, it just goes to show, I mean, we wouldn't be able to do this kind of work if it weren't for people like you paying attention to the environment around you. Yeah, and I guess I will, if you can advance the slide, please. I won't speak very long, but I will speak a bit to this vision that we share for the bioregion. The first slide here on the left is the Biodiversity Galiano project, kind of zoomed out. You can see those are observations from Galiano Island. And we're using iNaturalist as a tool, just like Bianca, to pull community observations in and then consolidating it all into a database where we can match up those observations with historical records that we've consolidated. And so that's the goal, is basically just to keep track of biodiversity and do so systematically with reference to what's been reported historically. And doing that, it's kind of a very basic framework, a pretty simple concept, really, just establishing a baseline and continuing to observe. But it's interesting how there's a lot of work to be done to develop systems for doing that systematically and doing it in a way that enables communities to really play a lead role in keeping track of their local biodiversity. And so folks like Bianca and others in the region are really playing really important lead roles, creating that capacity for communities to contribute in this way. It's just, there's so many, and it's, yeah, as Bianca shared, like many examples of really remarkable finds that are being made by people who don't necessarily hold any credentials, but they're really the ones in the best position to make that discovery just because they're in that place and paying attention. Yeah, the next slide here is zooming out even further from Galiano, just sort of situating myself in this and within the Salish Sea bioregion. And the next slide will show that. These are biodiversity projects that are active on iNaturalist. So we've just sort of started a collection project of the various projects in the region, and we've been sort of forming this federation of biodiversity projects, and each one is kind of realized to different capacities. And but I don't know, it's been 5 or 6 years, we've held this vision, and on the right you can see is this sort of gesture toward next steps of actually integrating the data from those areas, and not just the iNaturalist observations, but also the historical data. So on the right, you can see some of the project areas, not all of them, but some of the areas that are now connected through this network, including Vashon down at the very south end and Cortez up to the northwest. And the larger polygon there is the Howe Sound Biosphere region, which within Canada is one of the most recently recognized UNESCO World Biospheres. And I have a kind of interesting story because I graduated from my undergrad at Quest University in Squamish. And I actually had done my Keystone project on caribou, but was a bit depressed by the status of these caribou in Canada. So I ended up, instead of presenting on that work, I presented on the biodiversity project that started on Galiano, which is the very early stages. And a woman showed up by the name of Judith Home, who I didn't know at the time, but and I, not many people actually turned up, and so I was grateful that she did show. But I didn't really get to know her until sometime later, and we met during a naturalist outing on Galiano. She came out with a group, and she told me that she was inspired by the biodiversity project on Galiano, and she started her own. And since then, those data have informed the UNESCO World Biosphere application, which finally just this last year gained status for the bioregion. And I traveled up there earlier this year, and they have this challenge of maintaining this database that Judith has been curating. And she wants to pass it off to the biosphere in the long term. So they've asked me, and it turns out I've got now a bunch of funding for my PhD work in their basically conducting biodiversity assessments, managing the biodiversity data, but also creating systems for tracking biodiversity change. And so that's where you find me now. I just started my PhD at UBC. I'm really lucky to be working with Dr. Mary O'Connor and having, the opportunity to really focus my research on the science of the detection and attribution of biodiversity change. And this is going to give me the capacity over the next 4 years to really focus on this vision that we've had and work with Bianca and others throughout the region to synthesize really important historical biodiversity data sets, as well as all of these iNaturalist observations, and start sort of piecing together a bioregional picture of, the biodiversity. Relating it back again to Bianca's talk, I thought it was really neat. Just you look at just the scale of one island, how much habitat diversity there is, and these patterns that you can see at such a small scale. And in recent history, we've learned a lot about biodiversity at very broad regional scales, I'd say, but we're only recently do we have the tools to really start resolving a much higher resolution picture of biodiversity as it varies from place to place. And it's thanks to tools like these that are democratizing these practices. So that's all I have to share. Thanks very much for having me. If you have any questions for either of us, it'd be great.
Thank you, Andrew. That's great. So,
well, we do have some questions.
Yeah,
you guys are ready.
Yeah, we have some questions, and I'll put this up as our last slide, just a thank you to all our sponsors and partners throughout the years here. And we can take questions, both Andrew and I.
Well, let's start out with mine, since I'm the question asker. Will you use recordings of night sounds to get species you might not find, like frogs or insects, that you listen and say, oh, well, that's X, Y, and Z species that you didn't find physically, but you can hear them, kind of like the bats?
Mm-hmm. Yeah, so you can do that with, that's how our owling surveys are done, to detect owls, because at night you can't really see them too well. And so the birders just go out at night and listen for the owl calls and record them. And actually you can store audio on iNaturalist as well as photos. And so you can actually get a research grade observation from enough people listening to an audio and concurring that it's the actual species.
Oh, interesting. I have a question here. Out of the 10 years of Bioblitz, what's been the biggest surprise?
Ooh, gosh. I think just that firsthand experience. Even for me, who works in a lot of ecological, field work, just having that 24 hours to look in a certain area, the amount of life that's around you, it's hard to describe unless you actually go through a Bioblitz and see it. That's the most eye-opening thing, I think, for me. And then also just the human community that's created, around it. I had no idea the ripple effects of that. I think that was really surprising too.
Let's see here. When is the next Bioblitz? When is that going to happen? That's a big question.
Yeah, that's a good question. We're wrestling with that right now because we have a lot of data. Like, you saw a very simplified, clean version of what the actual mess looks like. And there, like, I'm sure Andrew understands this. They're just like, when you're dealing with a lot of these different observations and lists, and they come in all formats. And so we have I would like to do a lot of cleaning and really curating of the data that we have. And so that we're responsible to that before we go forward. So I don't know, maybe that'll take us a year. Maybe that'll take us another 2 years. I'm not sure, but we're certainly going to do a Bioblitz again. They're just so fun. And we've also wondered about, most of our Bioblitzes have been in the summertime. And so, should we do one in the winter? Because we would pick up completely different things. So the answer to that is I don't know when the next one is going to be. Maybe Andrew's got one. We can go to Galliano at some point.
Is there any part of the island that maybe is a microhabitat that you haven't explored yet that's maybe on your wish list to do a blitz on?
Oh yeah. Oh, I've got, yeah. And you know, you could talk to probably any Bioblitz participant and they'd have their next one. I mean, we haven't done Whispering Firs Bog yet. You know, that would be a really cool one. Bog environment. There are certain areas of the island, like large chunks that we haven't hit, like, you know, the southwest portion of the island. Camp South would be an amazing thing to look into more deeply. It'd be cool to do an underwater Bioblitz, like a scuba Bioblitz, because we're, you know, we do some snorkel surveys and we've done, you know, a few. I just think getting down a little farther into the depths would be really cool too. So,
well, how many islands in the Salish Sea do these Bioblitzes?
Andrew, you want to answer that? Do you have an idea?
What
are in that network?
It's hard to say just because like I said, some are loosely affiliated and really just doing Bioblitzes through iNaturalist, which I often say that the Bioblitz is never ending here on Galiano because people can continually contribute. I don't have an answer for you, to be honest. I know the San Juans, for example, there's a project encompassing the whole of all of those islands. So I'm not as aware of like independent projects on each of them, but some of them I've heard of down in the San Juan.
We've talked before, Andrew and I, about you know, getting a traveling band of people to go around and do Bioblitzes all over these different places. I think that would be so fun.
Oh, huh. Like have specialty in each group that would just kind of parachute in and do it and go on to the next island. Interesting. What species across the years has been the most abundant?
Well, we don't take abundance records, so I'd have to be like kind of qualitative about this, but I would say probably things like Douglas firs, sure. You know, salal, some of the things that you see. I'm sure we've gotten Douglas firs and salal and nettle in every single Bioblitz probably. But we don't take abundance records, so I can't tell you in terms of, you know, percent cover or number.
Okay. Well, does anyone else have any questions they'd like to put in the chat? For these, the brain trust goes away.
I think I saw a couple things here. There's a comment from Ren, who was one of our mushroom team leads, fungi team leads, and he says the biggest surprise for him would be the number of islanders who turn out for this great scientific effort. And that's true. Like it does attract, we get about 100 people to 200 people each time.
Yeah, I mean 900 amazing. Overall. Geez,
yeah, I mean that says a lot about humanity. You know, it's pretty cool, and I love that.
Do you have a lot of repeat people that come every year, or?
Yeah, we do. Yep, we have a lot of people who come. A lot of our team leads have been there for most of the years. Jim Evans, Raina, Alan. A lot of people. It's pretty cool.
I have one more question here. For someone new to the region, what is the best way to get involved in the Nature Center and learn about the species on the island, aside from participating in the next Bioblitz?
Yeah, that's a great question. You can go to our website and we have an events tab, and we put up we do a lot of community science, not just the Bioblitz. So we put up on our events calendar when certain things are happening. So we do forage fish surveys, we do salmon watching, we do beach surveys for other things in the summers. So we have a lot of stuff going on, and just go to our events calendar. We also have a contact from the webpage that you can just email us and let us know what your interests are, and we can plug you into the different things going on.
Okay. Thank you very much, Elsa. I think I can hand it over to you.
Thank you. Thank you, Chris. Thank you, Bianca. And thank you, Andrew, for coming from so far away to join us. And thanks to everyone for joining us this evening. We had a lot of people here. This was a wonderful presentation. It's just so exciting. You're going to get a lot more people at the next Bioblitz, even if you do it in the winter. I bet. And you can join.
Yeah.
Please, everybody come see Natural Wonder at the museum. We love having the Vashon Nature Center and Bianca as our partner. And because we're partners and you're partners with everybody in this community. And that's how we create these wonderful exhibits. So just want to say thank you to everybody and have a nice evening.
Thanks,
Goodbye.
everybody. Nice to see you. Thank you, Andrew.
Thank you, Andrew.